Good morning, everybody. Great to see so many people here and taking the time to come and learn about some of these cool companies. And I mean, some of the companies presenting this morning, just some really, really amazing, fantastic, cool technology. So we're really lucky to live in a time like this where there are just so many phenomenal advances. I'm going to take things back a little bit, and I'm going to tell you a little bit about Antion Biosciences, and we're a substantially earlier company. We're a spin-out company based out of Geneva, and really, when we started out, we took a look around at the best technologies out there and thinking about allogeneic cell therapies, which is really where we're focused on initially, and then moving into in vivo therapies. And we had a look at editing as the way to move forward for allogeneic cells, and it's a phenomenal technology. Doesn't matter which editing technology you use, it's incredibly powerful, and it's changed the way we develop therapies. But as we looked a little bit deeper and deeper, and we realized, and we spoke to more people, we realized that in order to create truly powerful, truly effective, truly persistent, and truly safe allogeneic therapies, you need to make more modifications to the cell than we initially thought we had to. So it's not just taking out the TCR, there are a few other things we need to do. And so we really sat down and thought, how can we do this better? And this allowed us to come up with our MyCAR platform that allows us to do real multiplexing, but still have very effective, powerful cells. So we all know at the moment the challenges that are associated with true multiplexing. You know, we've had the allogeneic, the autologous therapies where just a CAR is put in, and we've had phenomenal results. These are really amazing therapies. But if we want to bring the costs down, we want to get into those bigger patient populations, we need to think more about how do we do this on a more regular basis with allogeneic cells as one option. And it doesn't matter where you're getting your allogeneic cells from. They could be iPSCs, they could be donors, they could be a variety of spaces. And so we have to start thinking about, right, first of all, we have to take out the TCR, and the FDA and everyone agrees with that. But then people started thinking about, right, persistence. So maybe it's HLA class 1. And so editing, as it is, binary, it's either there or it's not. So that had to get taken out. But then all of a sudden, the NK cells come in when you put your cells in. And so, now we've got to put in a minor HLA again. So all of a sudden, you've got 3 manipulations. Then you start thinking about, do we need any inhibitory receptors? And we've seen from some of the other presentations already, they're particularly thinking about solid tumors. We may want checkpoint inhibitors. We may want other mechanisms and other edits in there. And so things start getting very complex very quickly. The result of this is, as you're probably all aware, the more edits you do, each edit is an action in and of itself, and it has its own consequences. The more edits you do, the more you tire out your cells, the more it ends up costing a lot of money, and that gets passed on to the consumer later on. So whilst editing is absolutely amazing, as we do more edits, we run into problems. So really what we've come up with is our mechanism called MyCAR, in which we're able to silence up to 6 different genes and add a CAR or a TCR, all within a single construct, in a single transduction step. So in one step and one transduction, we can do all of the engineering that is required. And this allows us to be very much more efficient. Now, in addition to that, where editing is purely binary, what we can also do is we can tune the degree of silencing for each of those 6 targets that we need. And so you think, well, why the heck would you want to do that? Well, the first thing is to think about what you might be doing when you take out your HLA class 1. Because if you're knocking out HLA class 1, you're having to knock in a minor HLA like an E or a G in order to prevent those cells being attacked by the NK cells. But this is where it gets interesting. We can actually design the silencing per target to decrease and to provide as much or as little silencing as we need. So we can silence by 50%. We can silence by 65, 75, it doesn't really matter. And the proof of concept is in HLA class 1. And what we've been able to show is that by actually silencing and knocking down HLA class 1 but not knocking it out, we can effectively turn it from a major HLA into a minor HLA. And so for those of you that are going to be joining us at ISCT in Paris next month, we're going to be showing data that actually shows the degree of silencing that we can do to ensure that those cells are not rejected, but they also are not taken out by the NK cells. So we've been able to find that sweet spot. And that's really, really exciting and interesting. So all of a sudden, we minimize the amount of editing and activity and engineering that has to happen to those cells. More importantly, when you start thinking about solid tumors, thinking about checkpoint inhibitors, this is where things start getting very interesting, that you don't want to knock out those checkpoint inhibitors entirely, but you want them knocked down substantially. So we have a number of real benefits over traditional gene editing. First of all is the tunability that I've already discussed. Our off-target effects, we're very, very fortunate in that the way we design them, we design each and every silencing construct from scratch, and it's all done in silico. And when it's designed in silico, it's tested against the entire genome, all in silico. So this allows us to be very, very certain of no off-target effects. And then we can still do all of the other required testing, RNA-seq and things like that, but that all becomes confirmatory. We have no toxicity because we are using the normal natural pathways, and so we don't clog up any of the systems within the space. And we've also had very, very high efficiency. At the moment, we're looking at, on average, 80 to 96% efficiency. And remember, we only do one transduction step, so we only have that one shot, and everything is in within that single shot. So this allows us to be very, very powerful and also to iterate very rapidly. Because we do all of our design and initial testing in silico, we can iterate very rapidly and we can test very, very quickly. So to show proof of concept, and this is really what we focused on last year, this is some data that we presented at ISCT and ASGCT. You can see the construct over there on the left-hand side of the slide. So we started off with a CD19 CAR. This was the same one that's used in Kymriah, so we kept that nice and standardized. And then you can see the 6 targets that we silenced there. Now, this is not a therapeutic construct. It's a bit of a Frankenstein construct, but it shows some cool things. So we started off, as you can see, CD52 for the lymphodepletion resistance, TIGIT, and a number of other receptors that we knocked out. Critically, CD3 zeta for the TCR was the furthest away from the promoter and enhancer. And that was done on purpose because that's the one that obviously we have to pay the most attention to and the FDA likes. And so we put that the furthest away to make our lives as interesting as possible. And so what we were able to show, and this is a very busy slide, and for that I apologize, you can see each of the different constructs and the degree of silencing. What we did here is we looked at the silencing over 29 days. This was multiple stimulations, so we really pushed these cells very, very hard. And if you look at the plots up at the top, the gray histogram is where there's no expression. The orange is where there's full expression. The teal color coming down is where it's just a single silencing construct on its own. And then the purple is as it's part of a multiplexed construct. So you can see that we get very little difference from just a construct on its own versus as part of 6 constructs together. Now, potentially we can go more than 6, and my CTO is desperate to get to 10, but it's all about focus, and it's all about, we probably don't need more than 6 at the moment. And so you can see on the bottom the MFIs. We were able to get all of those effectively right down, so functionally very nicely silenced. Now, that's all well and good if you can do multiplex silencing, but what about the CAR in the construct? And if you've got bleed over from the silencing to the CAR, that's not much good. And so what we were able to do is we were able to show in killing assays, and here you can see on a 1:1, 1:3, and a 1:9, and that's effector to target ratio. And so what you could see, the orange line is the untransduced. These are plain CAR T cells. The next one, that sort of teal color, those are effectively an autologous cell, an autologous CAR T, and then the purple is with a multiplex silencing also. And so on a 1:1 and a 1:3, we get pretty much the same efficacy. Interestingly enough, we got better efficacy in the 1:9, so 1 effector to 9 targets. I grant you it was only 3 donors, Read from it what you will, but we did get better effect, and so that's something we didn't expect and we're trying to understand at the moment. So what are we going to do with this? Well, our first indication that we're moving into is in T-cell malignancies. And so these include non-Hodgkin's lymphoma, T-cell non-Hodgkin's lymphoma, T-cell AML, and peripheral T-cell lymphomas. And those of you that are familiar with the space will realize, one of the problems with an autologous therapy is you might have contamination. And so we solve this with allogeneic cells. The other one is fratricide. If you armor and you put T-cell CARs onto T-cells, they're likely to kill each other if you target them towards each other. And so our multiplexing allows us to really prevent that completely by silencing the effects that we're going after. And then T-cell aplasia, we've got a number of ways of solving that issue. So moving forward, this is what our construct will look like. We have a dual CAR with CD5 and CD7, and that gets us about 85 to 90% of T-cell malignancies. We're silencing the TCR. We knock down, but not out, the HLA class 1, and then we also functionally silence CD5 and CD7 to prevent fratricide. So that gives us a really nice, strong construct. At the moment, we're deciding, do we need to put anything else in? Obviously, there's a suicide switch in there, which I think is absolutely important at this stage, given the power of the cells. It's a relatively small group of patients, and the real total addressable population is about 6,500 patients per annum in the US and EU. So it just allows us to really show the benefit, but then move on to other indications which we have already in development. So really, where the company is at the moment, as Jeff was mentioning also, like a lot of other people, we're out raising funds at the moment. We've got a collaboration with Allogene, where they have done proof of concept on our platform, and we are generating their next generation platform, allo platform for them. And so that's been very cool to work with them and really learn from them. So at the moment, we are taking our first construct forward into pre-IND testing and validation as we move forward in our planning. I'm not going to bore you with all the pictures of everyone because that's kind of sad, and I know the time is tight, so I want to give people a chance for questions and for the other groups to come on. So really, just as a summary, the Antion technology allows us really to silence 6 targets simultaneously in any cell type. And this is something I haven't mentioned to date. All of the data we've shown here is on T cells, but we've also shown this work and this capability in CD34 cells, in NK cells, in gamma delta T cells, and we've also done it in iPSCs, and we are in the process of showing it in macrophages at the moment also. So it gives us an enormous flexibility for different cell types out there. The benefit from cost, from an efficiency, from a regulatory complexity and quality is it's all single step. Single transduction, one shot, and your cells are created. We have this ability to tune, and we can tune per target, which is really, really exciting and gives us enormous flexibility. And with all of this, as you can imagine, there's an enormous amount of CMC optimization and the ability to bring our cost of goods down quite dramatically and hopefully add real value to patients and the payers. So that's it from me. Hopefully you found this interesting, and anyone that's interested, I'd love to catch up afterwards. So thanks very much for your time.